All right, welcome back to the second PCR lecture thing. Um, Due to popular demand, we have another one. All right, and thank you to all of our generous supporters on Patreon. You made this happen, everyone. Right, we got another couple minutes here to talk to you about a different version of PCR called qPCR. And so let's jump right into it here. So remember from the other video, the whole reason why we're doing PCR here is we are trying to, in this case, find any gene, right? Any gene that we know of, we can try to find. And so we're going to focus on, you can do any of these four things. We're going to focus specifically on trying to find viral and bacterial DNA here because that's really what it's all about. Right now, obviously very relevant. You're probably sick of hearing about it, but this COVID-19 is our reality for now. So let's kind of focus on that a little bit. And how would we test somebody for it? That's really what this is all about, right? So after they isolate, they swab your nose and they get your cells from your nose and whatnot, or even like some mucus or secretions or something like that from the nose. And in addition to your cells, there's also virus particles in there too, and so they can extract the viral DNA from that, and then they can run PCR on it. Now, you might remember from the virus POGL that you did, the COVID virus or coronavirus, no coronavirus is a DNA virus, it's actually an RNA virus. And so how do we get DNA from RNA? You might recall we have to use that reverse transcriptase enzyme. So there are viruses that have that enzyme where they can go backwards and you can actually go and take RNA and turn it into DNA. And so that's what we'd have to do with the coronavirus. So not only do they isolate the viruses, they got to blow them up, release the RNA, and then they got to reverse transcribe it and actually turn that RNA into DNA. And then from that point on, then they can do qPCR, right? So that's kind of what they got to do there. Real quick overview of what they'd have to do. So, so let's take a look here, qPCR, right? So all those four things we talked about can be done with qPCR, and the Q stands for quantitative. So it allows you to quantify how much DNA you've made. So, the whole point of PCR, again, remember, is to make a whole lot of DNA. And qPCR is a way for you to actually be like, well, yeah, rather than, I made a whole bunch of DNA, like, okay, well, literally, how much did you make? I made this much. And you can determine how much you make by these fluorescent probes. So that's how a lot of molecular biology stuff is done, is through fluorescent probes, because there's so many, all these chemicals and stuff, you really can't see them. And so we have to add probes to allow us to see them, because we can see pretty fluorescent things. Like you can see down here, I'm going to scroll this a little bit. We have these two pictures here. This is a turtle where different tissue has been stained with different fluorescent colors. We commonly use green and also red. And then sometimes where the two come together, you see yellow and things like that. But so yeah, the turtle's tissue has been stained different colors, and so has this anglerfish here. We've got red and green, again, the common one. So we can see fluorescent stuff. We can't see DNA so much. And so we use fluorescent tags to help us see the DNA and allow us to know that, yeah, it's there, or no, it's not. So that's what these probes do. So we can quantify how much DNA has been made with these probes, and here are the two probes that we use. You don't have to know the names of them or anything like that, but just realize here's how they work. Like this one here, this cyber, you get these little green dots which actually bind to double-stranded DNA. So every time we copy DNA using PCR, remember we're copying and making a double-stranded piece, these little green probes get stuck in that DNA. And so every time the DNA is made, these probes are there, and the more DNA is made, the more probes show up, and the brighter the color. Then there's this TACman stuff, which, remember TAC from TAC polymerase, that's kind of where that came from. And what the TACman does is, the way this one works is you attach the probe to your primers, and then those primers stick to your DNA, and then as soon as that DNA gets copied, this primer gets when the polymerase attaches to the primer and copies the DNA, this fluorescent tag gets activated. So you can see in this picture here, it's dull, and then over here, it's bright after it's been copied. So no copy, it's dull, copied, it's bright. And then same thing with over here. We copy the DNA and the probes are bright. That's really what you need to know, is the idea that the more we copy the DNA, the more fluorescence we have, and the more fluorescence we have, the more product has been made. Right? That's kind of what we got going down here. So these probes are activated every time we copy DNA, and more copies equals more fluorescence. And so the level of fluorescence confirms whether or denies whether or not you have the DNA you're looking for. All right. And so here's those two pictures that you saw on the a few slides before. And so I tried to kind of like, add probes to this. So like, imagine we have these green fluorescent probes here, and this is my initial DNA piece down here. Every time I copy it, the probe gets added to that DNA. Right? And so notice how I've got 1, 2, 3, 4, 5, 6, 7, 8 copies already. So now I have 8 probes. That's like 8 light bulbs shining, right? What's going to be brighter? 1 light bulb shining, 2 light bulbs, or 8 light bulbs, right? So 8 light bulbs is going to be a lot brighter. And so 8 probes is a lot brighter. And the way that shows up on your graph is you're going to get a curve that's really, really steep. So you got this green curve here. This green curve is the steepest out of all of them. It's the steepest the curve means you have more DNA. And why do you have more DNA? Well, because it was there, and we copied it with PCR. And so in this situation over here, I have this kind of blue colored probe where we only have a small amount of initial DNA. And so what that's going to turn into is you're going to have this kind of curve where you're going to have like pretty much hardly anything. It's going to be a lot lower than all the other ones. You might even end up with something that's way lower or something that doesn't even show up. So, I mean, maybe you'd end up with, a curve that's something like this, like it just doesn't even get there, right? And so if you have pretty much, this little blue line would be pretty much like no result, or if you have like a low result, because you're always going to be comparing them to other things. So if you have a low result or a no result, then you don't really have a lot of DNA. And so the scientists or doctors or whomever would say that's a negative result. So if they were to go ahead and swab you and test you for this coronavirus, they would, isolate the RNA, turn it into DNA, do PCR on the DNA, and add these probes. And the more times you copy the DNA, the more probes you have, the more fluorescence you have, the brighter it is. And then that curve shows up here, and this result will literally get produced by the machine. If you look at the video on the PCR virtual lab, they tour this lab in Minnesota, and it literally spits out a graph that it's way more busy than this. This is just a simplified one. But bottom line is the steeper the curve, the more DNA you have, so that's a positive result. If the curve is less steep or really not even there at all, like in this little blue one that I drew, that's what we would call a negative result, which means you don't have the virus in this case or whatever it is we happen to be testing for. So again, you would get a similar graph like this, not just for coronavirus testing, but you'd also get it for genetic testing or any of that type of stuff. Like if you were to test somebody to see if they had cystic fibrosis or something like that, you would isolate their DNA, run primers on the cystic fibrosis gene, and if you end up with a green curve or a red curve, that means you're positive for the cystic fibrosis gene, the version of the gene that gives you that disease. But if you have one of these blue results, we would say you're negative and you don't have it. So that's something to keep to be aware of. Again, really, really relevant right now. I'm going to run through, I got this virtual lab on qPCR that I found. I'm going to work through that later and see if it's any good. Maybe we'll do that as well. I'm not really sure how testing is going to be and assessments and all that sort of stuff, but if we end up, getting back together and stuff, remember how we do those tests where there's data and writing and all that fun stuff, you might end up seeing a curve like this or, a graph like this with some curves, and so be able to interpret these curves and understand which curve means, yes, I have DNA, which curve means no, I don't have any DNA, right? And so again, steep curve means lots of fluorescence, lots of DNA. Shallow curve or no curve at all means not a lot of DNA, not a lot of fluorescence. So that'd be like what we call a negative result, right? So my not so long video ended up being 10 minutes, but whatever. You haven't heard me talk for a while, so I'm sure you just love hearing it, right? You couldn't wait to listen to it. But at least the nice thing about the videos is you can always pause me, mute me, or just walk away, which you can't always do in class. So at least you got that going for you, right? All right, I guess that's it. Hope you enjoyed it. Again, if you got questions, please feel free to reach out and we'll go from there. All right. Thank you. Be well. We'll see you later.